warahmatullahi wabarakatuh. So we are from Group 7. I am the group leader. My name is Siti Nur Iman Hidayu binti Syahrini. Okay, next.
My name is Nur Alia Tasni binti Mat Daud. Okay, next.
My name is Nur Nabila binti Adnan.
Assalamualaikum warahmatullahi wabarakatuh. My name is Iya Iskandar bin Azrin.
So today, we want to present our notes. Okay, let's start with me. Wait a second. Sorry, there's some problem.
Tak apa.
Okay, I will start presenting now. Can you see my slide?
Yes.
Okay. Today I will present about carbohydrates. I will explain to all of you about their structures and also their functions. Okay, carbohydrates. Before we start, let me give some definition about carbohydrates. So, carbohydrate is a biomolecule consisting of carbon, hydrogen, and oxygen. So, atoms. So, usually, carbohydrates have hydrogen, oxygen atom with ratio of 2:1. So, hydrogen 2 and oxygen atom 1. That is the ratio. Next, there are 4 classification of carbohydrate. First is monosaccharide. Monosaccharide is the basic of carbohydrates. So next, disaccharide, oligosaccharides, and polysaccharides. So in the table we can conclude some information about the categories of carbohydrates. So monosaccharide is formed by one sugar molecule. Disaccharide from two sugar molecule. Oligosaccharide is 2 to 10 sugar molecule, and polysaccharide is 10 or more sugar molecule. Okay, let's move on to monosaccharide. Okay, monosaccharide, the most basic forms of carbohydrate. So it has we can call it a simple sugar also, because monosaccharide cannot be further hydrolyzed. It means it cannot. It is the basic unit of carbohydrates. So the general formula you can see it is CnH2ON or NH2NON. Okay, next. Examples of monosaccharide is glucose, fructose, and galactose. Okay. The structure of monosaccharide. All monosaccharide have two general formula, which designate a central carbon molecule bonded to two hydrogen and one oxygen. So the oxygen will also bond to a hydrogen, creating a hydroxyl group. Because carbon form for bonds. Several of these carbon molecules can bond together. One of the carbons in the chain will form a double bond with an oxygen, which is called a carbonyl. So this is the example of glucose structure. This is the function of monosaccharide. So monosaccharide function to produce and store energy. As we know, carbohydrate can supply energy for us to doing activities all day long. So it also to form long fiber, which can be used as a form of cellular structure. Some bacteria also can produce similar cell walls using slightly different polysaccharides, all from smaller monosaccharide. So polysaccharide is formed by combining two smaller monosaccharide. So next we can move on to disaccharide. Disaccharide, also can call double sugar, is the molecule formed by two monosaccharide. So two monosaccharide combine and become disaccharide. So disaccharide are formed by dehydration reaction, in which a total of one water molecule is removed from the two monosaccharides. Example of disaccharide is sucrose, maltose, and lactose. Structure of disaccharide. The structure of disaccharide is much similar to of monosaccharide. So it consists of multiple hydroxyl group 2 and a functional group that could be aldehyde or a ketone. The only difference is the presence of glycosidic bridge connecting the two molecules. Function of disaccharide is found in many food, often added as a sweeteners. When disaccharide like sucrose are digested, they are broken down into their simple sugar and used for energy. So disaccharide can be broken down through process of hydrolyzation. So it can become monosaccharide back. Plants store energy in the form of disaccharide like sucrose and also used for transporting nutrients in the flow. Next is oligosaccharide. It is comprised 3 to 10 monosaccharide. So 3 to 10 monosaccharide combine and form oligosaccharide. So a large number of oligosaccharide have been prepared by partially breaking down more complex carbohydrate that is polysaccharide. We will learn polysaccharide after this. So examples of oligosaccharide is raffinose and stachyose. Okay, structure of oligosaccharide. So oligosaccharide comprise 3 to 10 monosaccharide, and they were linked together mostly by O-glycosidic bond through condensation reaction between anomeric carbon of monosaccharide and the other. This is the example of O-glycosidic bond. So they also can form an glycosidic linkage under certain atmosphere. So this is the example of N-glycosidic bond. So next, function of oligosaccharide, uses for cell recognition and cell binding, used by beneficial colony bacteria in large intestine to break down oligosaccharide to absorbable nutrients, act as soluble fiber which may help prevent constipation. So lastly is polysaccharide. So polysaccharide is the most complex of carbohydrate. So a long chain carbohydrate made of monosaccharide and connected together via glycosidic bonds to form the polysaccharide. So examples of polysaccharide is starch, glycogen, and cellulose. Structure of polysaccharide is complex biomicromolecules. Type of the polysaccharide is glycosidic bond. So various monosaccharides linked with glycosidic bond made up of polysaccharide. So this is example of starch that is have a glycosidic bond. So the function of polysaccharide is common source of energy, provide important dietary elements for human, and lastly, enhance digestion. That's all from me. Next, we move on to Nabila. Alright?
Okay. Nampak tak?
Nampak.
Okay. Nucleic acid is linear polymer molecule. These are not brains composed of subunits or nucleotides. It's complex molecule has carbon, hydrogen, oxygen, phosphorus, nitrogen. Nucleic acid, biological molecule as genetic material, plays an important role in the storage of genetic information and protein synthesis. To form enzymes, hormones, protein, and subunits to DNA and RNA. Nucleic acid structure the main information carrying molecules of the cell, and by directing the process of protein synthesis, they determine the inherent characteristic of every living thing. Nucleic acid structure along nucleotide subunit chain is called polynucleotide. Nucleotide monomer of nucleic acid. Category of nucleic acid, deoxyribonucleic DNA and ribonucleic RNA. The most basic structure of RNA and DNA bond between monomers, nucleotide bond, monomer structure or one nucleic unit have three components. First, phosphate group. Second, a 5-carbon sugar, and third, nitrogenous base. Phosphate group gives acidic properties to nucleic acid. A 5-carbon sugar, DNA and RNA consist of different source sugar. A nitrogenous base, two types of nitrogenous base consisting of purine and pyrimidine. Okay, ribonucleic acid, RNA, short chains of polynucleotides found in the cytoplasm, ribosome, and nucleus. RNA, named based on ribose sugar, is one of the components of its nucleotide. Base of nitrogen, RNA, cytosina, adenina, guanina, and uracil. RNA structure, single G. RNA molecules are synthesized from DNA and are very important in the process of protein synthesis in the cytoplasm. Diosiribonucleic acid, DNA, consists of two polynucleotide chains twisted into a double helix found in the nucleus of eukaryotic cell and the nucleus of cytoplasm of prokaryotic cells. Named based on the diacylrose sugar, which is one of the components in its nucleotide base of nitrogen, DNA, adenine, guanine, cytosine, and thymine. DNA molecules are made up of two nucleotide chains that form a double helical structure. This is summary of difference between DNA and RNA. DNA consists of two separate polynucleotide chains arranged in parallel and long. And RNA consists of a short chain of polynucleotides. DNA double helix and RNA single chain. DNA have diacylribose and RNA ribose. I think that's all. Okay, thank you.
Next, we move on to Yen.
Okay. Assalamualaikum. Boleh dengar tak?
Boleh?
Okay. Okay. Assalamualaikum warahmatullahi wabarakatuh. I will be presenting the topics on lipids. So the first one, definition of lipids. Lipids are group of organic compounds that are insoluble in water and also soluble in non-polar organic solvents. They naturally occur in plants, in animals, microorganisms, and also used as cell membrane components, energy storage molecules, insulation, and hormones. Lipids can be broken down into two parts. The first one is glycerides, and the second one is fatty acids. The first part, glycerides, can also be broken down into smaller components. The first one being phosphoglycerides, the second one being neutral glycerides. Now, for the second part, that is fatty acids, also has two components. The first one is saturated fatty acids, and the second one is unsaturated fatty acids. Now, there are also two parts that came from lipids that are different than fatty acids and glycerides. The first one would be complex lipids, and the other one would be non-glyceride lipids. Now, for the complex lipids, it could be turned into a much simpler component, and then the lipoproteins. And for the non-glyceride lipids, it could be broken down into three components. The first one is waxes. The first one being waxes, the second one being sphingolipids, and the third one is steroids. Now, for the sphingolipids, it could be broken down into two more smaller components. The first one is sphingomyelids, and the second one is glycolipids. And those are the components that make up what lipid is. Lipid is a general name, but if you want to specify what lipid is, those are the names or components in lipids. They are considered as lipids. Now, I'm going to explain about the properties of lipids. The first one is lipids. It could exist in liquids or non-crystalline solids, only at room temperature. The second one is pure fats and oil are colorless and odorless and tasteless. The third one is that they are energy-rich organic molecules. The fourth one is insoluble in water. The fifth one is soluble in organic solvents like alcohols, chloroform, acetone, and benzene. The sixth one is that lipids has no ionic charges. The seventh one is that lipids considered as solid triglycerols. They have high proportions of saturated fatty acids. And the final one being liquid triglycerols, which is oil, have high proportions of unsaturated fatty acids. Okay, so the next part, I will go into the structure of lipids. And lipids are made of the elements carbon, hydrogen, and oxygen. But they also have much lower proportions of water than other molecules such as carbohydrates. The second part is that unlike polysaccharides and proteins, lipids are not polymers. They lack a repeating monomeric unit. The third part, lipids are made from two molecules. The first one is glycerol. The second one is fatty acids. The fourth part, a glycerol molecule is made up of 3 carbon atoms with hydroxyl group attached to the glycerol molecule, and it also has hydrogen atoms occupying the remaining position in that molecule. And the fifth part would be fatty acid is that if every bond that exists is made with hydrogen atom. And another part is saturated fatty acids do contain CC bonds. Monosaturated fatty acids have 1 CC bond, and polyunsaturated have more than 1 CC bond. This part would be the structure of triglyceric lipids, consisting of only 1 glycerol molecule that bonded with 3 fatty acids. The bonds between these molecules are covalent and are called esters. Triglycerols are formed during condensation reaction. And the final part of the structure of triglycerides is that the charges are distributed evenly on the molecules so that hydrogen bonds do not form with water molecules, making them insoluble in water, because the hydrogen element cannot bond with water. The next part would be classification of lipids. And the first part, the classification of lipids, is that there is simple lipids, and simple lipids will be broken down into 2 parts. The first one is fats, which yield fatty acids and glycerol upon hydrolysis. The second part is we yield four compounds. They are called triacylglycerols, because they are esters composed of three fatty acids joined together with glycerol and trihydroxy alcohol. Now, the difference is on the basis of their physical state at room temperature. It is customary to call a lipid fat if it is solid at 25 degrees Celsius and or it is liquid at the same temperature. And these differences, the differences in the melting point of this ingredient, they represent the differences in the degree of the unsaturation of the constituent fatty acids. For the waxes, wax is an ester of long-chain alcohol, usually in monohydroxy and fatty acid. And alcohols normally found in waxes have chains of the order of 12 to 34 carbon atoms in length. Compound of lipids. The first one is phospholipids, which give fatty acids, glycerol, ST-amino, and nitrogen containing alcohol upon hydrolysis. Phospholipids may be glycerophospholipids or sphingophospholipids. This alcohol group is sphingosine, and you could also derive lipids during hydrolysis, and the product of hydrolysis is called derived lipids. Now, the functions of that it could be used to store energy inside a cell, and that cell could be inside a plant or an animal or a human body, or even other microorganisms. B also needs to store excess lipids. Other function for lipids is that it would make up the biological membranes. Lipids are also used for insulation. It is used for protection, such as in plant, where plant would use lipid to protect the plant leaves. And lipid is also used for buoyancy, large amount of lipid or fat under their skin, and it helps on water. And other use for lipids is that it could be used as hormones inside not just human bodies but also in plants and animals, and they act as the structural component of the body, and it also provides hydrophobic barrier that permits the partitioning aqueous contents of the cell and subcellular structures. And lipids are primary sources of energy in animals and humans, and high lipid containing seeds. Lipids are also used as activators of enzymes such as glucose and phosphates, as it require phosphatidylcholine for activation. And that's about all the things that I present. Thank you.
Okay. Can you see my slide?
Yes.
Okay. Okay, I will start present about protein. Protein contains the element carbon, hydrogen, oxygen, and nitrogen. Some proteins contain sulfur and phosphorus. All proteins are made up of one or more monomers or unit known as acid amino. Dipeptides are formed when two amino acid molecules are bound together by a single peptide bond through condensation. Dipeptides can be broken down into amino acid through hydrolysis. Amino acid plus amino acid by condensation equal to dipeptide plus water. Dipeptide plus water through hydrolysis equal to amino acid plus amino acid. Polypeptides, proteins or polypeptide hydrolyzed to amino acid by digestive enzyme will be absorbed into the blood. Next, these amino acids are used to build different protein molecules needed by the body. Polypeptides plus water by hydrolysis equal to dipeptide or amino acid. Okay, type of amino acid. All 20 types of amino acid can be divided into two groups, namely essential amino acids and unnecessary amino acid. Essential amino acids are amino acids that cannot be synthesized by the body. An example of an essential amino acid is leucine. Animal protein contains all the amino acids needs to be known as first-class protein. Unnecessary amino acids are amino acids that can be synthesized by the body. These amino acids are formed by other amino acids. There are 11 types of unnecessary amino acids. Plant proteins thus do not contain all the essential amino acids. This protein is known as second-class protein. Okay, protein structure. Primary structure. The primary structure of a protein refers to the linear sequence of amino acids in a polypeptide chain. The genetic code carried by the DNA molecule in the nucleus is an example of a primary structure protein. Secondary structure. Secondary structure refers to a polypeptide chain that twists to form a helix, alpha, spiral, or a polypeptide that fold into a beta-shaped sheet. Both of the helical structure and the fragile pieces are held together by hydrogen bonds. Okay, third is tertiary structure. Tertiary structure refers to the way a helix alpha chain or a folding beta sheet form a solid, three-dimensional globular protein complex. Enzymes, hormones, antibodies, and proteins are examples of complex proteins of tertiary structure. Okay, the last is quaternary structure. Quaternary structure refers to the combination of two or more tertiary structure, polypeptide chains forming one complex and large protein molecule. Hemoglobin has a quaternary structure. Okay, that's all for me. Thank you.
Okay. I think that's all from us. Okay. Thank you. Okay, dah boleh